signaling. Let's first understand why phytohormones are so important in plants development. Of course, we know about not only my lectures that are chemical messengers that regulate almost every aspect of plant growth and development. They control key processes, and these hormones work in a coordinated manner and of course coordinated or interact with other phytohormones. But why should we model phytohormone signaling? There are two primary reasons. The first, of course, understanding complex pathways, and the second, predicting outcomes of genetics or environmental changes. Phytohormone signaling is highly complex, involving multiple feedback loops, interactions between different phytohormones, and responses that vary depending on the developmental stage or environmental conditions. By modeling these pathways, we can systematically analyze and predict how phytohormone regulate plant growth. What about the predictive? Computational models allow us to stimulate the effects of genetic modification, such as gene knockout or overexpression, or environmental factors too, such as drought or extremely low or high temperatures. This is particularly valuable for optimizing crop traits and developing stress-resistant plant varieties. Let's explore why modeling this signaling is crucial. We know that phytohormone signaling is not a linear process. It's a highly intricate network that involves multiple feedback loops that regulate hormone levels and responses. The second is a crosstalk between different phytohormones influencing multiple development processes. And the last is context-dependent signaling, meaning that the same hormones can have different effects depending on tissue type, developmental stage, or environmental conditions. Because of this complexity, traditional experimental approaches alone are often insufficient to fully map out hormone interactions. This is where a computational model becomes a powerful tool. By developing mathematical and computational models, we can systematically analyze how different components of hormone signaling interact. The second, identify key regulators that drive specific development or stress responses outcomes, and integrate data from multiple experiments to form a comprehensive picture of how phytohormone function. Example, in auxin-cytokine interactions, computational model help us understand how hormone gradients influence root and shoot development. It's a typical physiological interaction between auxin and cytokine. It's typical even books. So, by stimulating this dynamics, we gain insight that would be difficult or even impossible to obtain through experiments alone. Ultimately, modeling allows us to make a prediction, test hypotheses, and refine our understanding of phytohormone signaling, all of which contribute to innovation in crop improvement, stress resilience, and prediction agriculture. Plants adapt to drought, temperature shift, and stress factors. Here we know why biotechnology enables genetics modification for improvement traits like drought resistance or enhancement yield. Computational model allows us to stimulate these conditions and predict how changes in hormone signaling affect plant development. For example, ABA signaling models predict stomatal responses to water stress, aiding in the development of drought-resistant crops. And the second, auxin transport models help optimize root growth and shoot branching patterns for better plant architecture. These predictions are sometimes save times and cost by reducing trial and error in experiments and improve crop trace by identifying optimal genetics modification and support precision breeding, enhancing hormone-driven plant responses. By integrating computational models with experimental data, we can design more and more resistant and productive crops. Now that we understand why modeling phytohormone signaling is crucial, let's focus on a specific example, brazinosteroids. I don't present the signal transduction of brazinosteroids in plants, but I would like to tell about modeling. Modeling brazinosteroid signaling help us understand how brazinosteroid receptor perceive signals and active downstream genes, and how brazinosteroids interacts with other phytohormones like auxin, gibberellins, cytokines, and others to coordinate plant growth, how brazinosteroid levels influence stress tolerance, especially against environmental challenges like drought or extreme temperatures. By stimulating brazinosteroid signaling networks, we can predict how changes in brazinosteroid activity affect plant traits, aiding in crop improvement. Why modeling phytohormone signaling network provides value insight, it comes with significant challenges. These signaling networks are highly complex, dynamic, and interconnected, made it accurate prediction difficult. Some key challenges are feedback loops and crosstalk, spatial and temporal variability, and limited experimental data. Many models require precision biochemical parameters that are difficult to obtain. So, addressing these challenges required advanced computational approaches and integration of high-resolution experimental data. Despite these difficulties, modeling remains a powerful tool for unraveling phytohormone function. One of the biggest challenges in modeling phytohormone signaling is its complexity. These pathways involve numerous genes, proteins, and feedback mechanism, creating in a highly dynamic system that is difficult to map and predict. The key complexities are following. multiple layers of regulation. That means that hormone activity is controlled at transcriptional, translational, and post-translational levels. The second, non-linear interactions. That means the effects of one hormone often depend on the presence of others, leading to unpredictable signaling outcomes. And the last, crosstalk between pathways. For example, auxin and cytokinins interact during organogenesis, making it difficult to isolate their individual roles. Because these factors, traditional experimental methods alone, cannot fully capture the system behavior, mathematical and computational models help simplify and stimulate this interaction, providing a clearer understanding of plant growth regulation. Another major challenge in modeling signaling is feedback regulation and crosstalk between different hormones. Feedback loops can be positive, amplifying the response, or negative, that means suppressing the response, which tune fine hormone action. For example, auxin transport is regulated by pin proteins, whose localization is influenced by auxin itself, creating a self-reinforcing loop. And the next, crosstalk occurs when different hormones influence each other's pathways. For example, abscisic acid and salicylic acid exhibit antagonistic crosstalk, where abscisic acid position draw response, while suppression salicylic acid driven defense mechanism. This interaction introduces non-linear and context dependence, making it difficult to isolate the effects of a single hormone aside. Once again, the same hormone can have different effects depending on its concentration, distribution, and developmental stage of a plant. We know everything about it. Key challenges include hormone gradients, tissue-specific responses, and time-dependent signaling. Hormone gradients, for example, auxin form localized maxima in root tips, directing meristematic activity and organ patterning. For the second, a hormone may promote growth in one tissue but inhibit it in others. Some hormones responses occur immediately, while others require long-term regulation. We know about these responses when you treated exogenously plants by phytohormones. To model these dynamics, researchers use special modeling approaches, such as partial differences equation is PDF as an abbreviation, which simulate hormone diffusion and distribution in plant tissue. These models are crucial for understanding how hormones coordinate growth across different parts. The next challenge in modeling is the lack of precise experimental data, which need to define model parameters accurately. Many models rely on quantitative values such as reaction rate for hormone biosynthesis and degradation, hormone transport coefficient, and binding efficient between receptor and signaling molecules. However, these values are often difficult to measure directly due to, firstly, hormone instability and the low concentration making detection challenging. The next, tissue-specific variability is required high-resolution techniques. And the last, complexity of interactions where multiple phytohormone influence each other dynamically. To improve parameters estimation, we are now integrating high throughout omics and data and advances imaging techniques to obtain more precise measurements. There are three main methods: experimental approaches, computational modeling, and integrative frameworks. The first, this include gene knockout studies, biochemical assays, and imaging techniques to observe hormone activity directly. In computational modeling, the mathematical and machine learning models help simulate hormone interactions, transport, and regulatory networks. At the last models, integrative frameworks, combine experimental data with computational tools, creating a more and more complete picture of phytohormone dynamics. Of course, each model has its strengths and limitations, but together they allow researchers to better understand and predict plant hormone behavior. Experimental methods play a crucial role, we know, but key techniques include genetical studies, biochemical assay, imaging techniques. Of course, each of these methods provides valuable insight, but they also have limitations. For instance, genetics modification may trigger compensatory mechanism, making it difficult to isolate specific effects. This is why integration experimental data with computational model is essential for building a complete picture of hormone signaling. Genetic studies are one of the most effective ways to understand phytohormone function by manipulating genes which are involved in hormone biosynthesis, transport, and signaling. There are some key approaches: gene knockouts and gene overexpression. The first, removing specific genes using techniques like CRISPR-Cas9, helps identify their role in hormone regulating. And gene overexpression, by increasing the expression of key genes, involve us to observe their effects on plant growth and development. For example, knockout studies of PIN transporters have revealed how auxin movement shape plant architecture. Similarly, overexpression cytokine-related genes has provided insight into shoot branching and meristem activity. While these approaches are powerful, they may not fully capture redundant or compensatory mechanism within signaling networks. This is why combining genetic studies with biochemical imaging techniques provides a more comprehensive understanding of phytohormone signaling. Biochemical assays are essential for measuring phytohormones level. We know about it. One of the most, of course, useful techniques which I presented today is LC-MS, which provides high sensitivity and specificity quantification of hormone fluctuation, etc., etc. We know everything from my previous lecture. Imaging techniques are powerful tools for visualizing hormone distribution and dynamics in plant tissues. These methods help us track where phytohormones accumulate, providing critical insight into their role in growth and development. One widely used approach is GFP tagging, where fluorescent markers are attached to hormone-related proteins or reporters. For example, the R5-GFP constructs allow real-time tracking of auxin maxima in root and shoot meristems. The next, fluorescence lifetime microscopy enables dynamic monitoring of hormone receptor interactions. While experimental approach provides direct insight into hormone signaling, computational modeling allows us to simulate, predict, and analyze complex hormone interactions that are difficult to study through experimental alone. Computational models use mathematical equations to represent biochemical reactions involved in hormone synthesis and degradation, signaling kinetics that regulate plant responses, and hormone transport and feedback loops, ensuring proper distribution and function. These models help identify critical control points in a signaling network and predict how genetics modification or environmental changes affect plant growth. However, computational modeling requires precise experimental data for validation. Integrating biochemical, genetic, and imaging data improves model accuracy, making it a powerful tool for crop optimization and stress adaptation research. Computational modeling of phytohormone signaling relies on various techniques to stimulate biochemical processes, spatial distribution, and regulatory interactions. There are four main approaches: mathematical and spatial modeling, machine learning and artificial intelligence techniques, agent-based modeling, and network-based modeling. The first technique use ordinary differential equation and partial differential equation to stimulate hormone synthesis, degradation, and movement across tissues. The second, machine learning and AI techniques, predict gene hormone interactions and signaling patterns using data-driven models such as neural networks. Agent-based modeling simulates individual cell behavior and how they collectively form tissues structures. Network-based modeling visualizes complex hormone, protein, and gene regulatory network, revealing key interactions in signaling pathways. What about ordinary different equations in mathematical modeling? Used to model hormone synthesis, degradation, and signaling kinetics over time. For example, LDE can predict how auxin levels or other, of course, hormones fluctuate in response to environmental stresses. Partial differential equation used for spatial modeling, incorporating hormone diffusion, tissue-specific localization, and gradient formation. This approach-based models help explain how phytohormone maxima guide root and shoot patterning, These models are essential for understanding how hormone concentration change in time and space, helping us predict growth patterns, stress responses, and developmental decision. Machine learning and AI techniques are enable data-driven prediction of gene hormone interaction signaling dynamics, prediction hormone regulated gene expression. The next application is a pattern recognition in a large data set. The next, optimizing crop traits. What's the mean? Predicting hormone regulating gene expression. Deep learning models analyze transcriptional data to identify hormone response genes. In the second pattern recognition in large data set, neutral networks detect hidden correlation between hormones, environmental stimuli, and growth patterns. The next, of course, AI helps predict how genetics modification affect hormone pathways, guiding precision breeding strategies. For example, machine learning has been used to predict auxin distribution in root under varying gravitational conditions, improving our understanding of gravitropism and plant adaptation. Despite its power, AI models often lack interpretability, requiring biological validation through experiments and traditional computational methods. The next, agent-based modeling, is a powerful technique for simulating how individual cells behave and interact in response to phytohormone signaling. Unlike traditional mathematical models, agent-based modeling treats each cell as an independent agent, responding to local hormone concentration and external stimuli. There are some advantages of this method. The first, capturing emergent behavior, modeling transport at a cellular level, and the last, studying experimental responses. Of course, by adjusting parameters, we can predict how plants respond to drought, light changes, or nutrient availability. This approach is particularly useful in modeling root meristem activity and shoot branching, helping researchers understand how hormones shape plant development. To fully understand hormone signaling, we must integrate frameworks which combine experimental data, computational modeling, and large-scale biological data sets. This approach provides a holistic view of how phytohormones regulate growth and development. There are some key components which integrate frameworks. Multi-omics approaches integrating transcriptomics, proteomics, and metabolomics to reveal hormone-regulating pathways. I can say not only but plant hormonomics, but we know that's a part of metabolomics. Bioinformatic tools, for example, platforms like the Arabidopsis EFP browser help visual gene expression changes in response to hormone. And the last, multi-hormone integration modeling. Understanding, for example, interaction between two hormones, crosstalk in cell expression of these hormone interacts in stress responses. By merging experimental computational data, integrated frameworks provide a more accurate and predictive models of phytohormone function, benefiting both basic research and agricultural application. One well-studied example is the crosstalk between auxin and gibberellin, which regulates cell elongation, division, and overall plant growth. There are, in model, there are some interactions. Gibberellin enhances auxin response. That means that this hormone promotes the expression of auxin responsive gene, facilitates cell expansion. And then auxin regulates gibberellin biosynthesis. That means auxin stimulates the production of gibberellin in rapidly growing tissues, ensuring coordinated development. And the last, tissue-specific effects. In short, auxin and gibberellin interactions drive stem elongation, while in root they help regulate meristem activity. We know that abscisic acid and salicylic acid play opposing roles in plant stress responses, making their interactions a key area of study in phytohormone signaling. Abscisic acid promotes abiotic stress tolerance, so it helps plants survive environmental stresses, environmental conditions by regulating stomatal growth and gene expression. Salicylic acid enhances biotic stress defense, so activates immune response against pathogens. Antagonistic regulation is under, for example, under draw conditions. Abscisic acid can suppress salicylic acid signaling to prioritize water conservation over pathogen defense. Conversely, during infection, salicylic acid reduces abscisic acid activity to enhance immune responses. What about next case, auxin signaling? Auxin transport models like canalization hypothesis explain how auxin flow directs patterning in leaves and root development. The next, of course, in auxin modeling, perception and signaling is important. I thought about receptor complex, mediates auxin responses, regulating transcriptional factors that drive growth. We know that important is gradient formation. PDE-based models simulate how auxin maxima influence cell division and differentiation, especially in meristem. The key feedback mechanism include, in this case, positive and negative feedback loops. What's the mean that auxin promotes the localization of PIN transporters, reinforcing its own directional flow, which is crucial for vein patterning and root development. But negative feedback loops mean that high auxin levels trigger degradation of auxin repressors, ensuring tie regulation of gene expression. Crosstalk of other hormones auxin interact with cytokinins or gibberellins, creating context-dependent growth responses. The next case of study, cytokine signaling. There are two key aspects. Two components signaling system, such as cytokinin receptor, for example, CRE1, activate histidine kinase, which transfer phosphate signal to response regulators. The next is should and root development. Computational models simulate how cytokinin gradients influence meristem activity and branching patterns. To fully understand phytohormone regulation, we may use multi-scale modeling, integrative all data from different biological studies, molecular, cellular, organismal levels. The first models focus on gene expression, protein interactions, and hormone biosynthesis. Cellular level mean that simulation track hormone transport, signaling cascades, and feedback loops within individual cells. What about organismal level? Models predict how phytohormone ingredients shape root growth, shoot branching, and stress responses. This approach bridges the gap between small-scale molecular interactions and the large-scale developmental patterns, helping us simulate plant responses under various conditions. What happens at the molecular level? Phytohormone bind to receptor, triggering a cascade of biochemical reactions. Transcriptional factors are activated, switching specific genes on or off to control plant responses. And enzymes synthesize, modify, and degrade phytohormones, adjusting their level based on the plant needs. Why model it? To simulate how hormone receptor interactions drive gene expression, to predict the effects of mutation on signaling pathways, and to understand how environmental factors influence phytohormone biosynthesis. By manipulating these molecular interactions, we can design experiments, optimize genetic modification, and develop stress-resilient plants. What about on the cellular level? What happened inside a cell? Hormones, we know that are synthesized and transported. Some move freely, while other phytohormone required carrier proteins. Signaling cascade begin when hormone, of course, bind to a receptor, serial molecular signals spread through the cell, and feedback loops regulate the process, ensuring hormone levels stay precise and balanced. Why model the cellular level? To simulate how hormones flow through individual cells and predict their interact on development. To understand how cells respond to fluctuation of hormone levels under stress or normal, of course, conditions. To study localized hormone distribution, how proteins, for example, direct auxin flow, pin protein in root or system tissue. From individual cells, let's zoom how phytohormone influence entire organ and coordinate plant growth. At this level, phytohormones act as global messengers coordinating all phytohormones, coordinating growth across different tissues and organs. What happened at the organismal level? Hormones create gradients directing root and shoot development. Environmental signal like light changes or draw trigger system-wide hormone responses. And hormones interact to balance growth and defense, ensuring the plant adapt to stress. Why model the whole plant? To simulate how hormone network shape over plant architecture, to predict how genetics modification impact plant form and function, and to optimize crop traits like improving drought resistance, for example, through abscisic acid regulation. Now that we have covered molecular, cellular, and organismal level, let's discuss about challenges bridging these scales in infinite model. Oops. What makes multi-scale modeling difficult? The first, different scale requires different data types, gene expression data at the molecular level, transport dynamic at the cellular level, and growth pattern at the organismal level, on 3 levels. Processes at the level affects higher levels, up predictable. For example, a small mutation in a hormone receptor can alter whole plant architecture. Computational complexity, simulating entire plants with high accuracy demands massive data sets and processing power. How tackle these challenges? For example, to create more accurate predictive models of plant growth, or to optimize plant traits for agriculture, improving resistance and productivity, or to design experimental efficiency, reducing the need for costly trial and error in genetical modification. So, despite the difficulties, advances in big data, AI, and high-resolution imaging are making multi-scale modeling more powerful than ever. Creating a model is just the first step. This is the real challenge which improves that it works. Validation is crucial to ensure that our prediction align with a real biological data. How do we validate models? By comparison with experimental data, by sensitivity analysis. We need key parameters, for example, hormone concentration, reactant rate, to see how much they affect the outcomes, and parameter estimation. That means we must have advanced techniques like Bayesian interference and machine learning, which help fine-tune models using real-world data set. Why is validation important? Because it increases confidence in model prediction, identify weakness, and ensures practical application. So, validated model can be used to design stress-resistant crops and optimize growth conditions. Model validation is an ongoing process. The more accurate the model, the more powerful its predictions. No model is useful unless it matches real-world observation. How is done? By simulated hormone gradients, by gene expression level predicted by models, and predicted hormone response, which are tested under controlled laboratory conditions. Why is it important? Because it's ensured that models accurately represent biological system. It defines this appearance that help refine models and builds trust in computational predictions, make them more useful for real application. Even the experimental validation model must be robust and adaptable. This is where sensitivity analysis play a key role. This is where sensitivity analysis come in. How does it work, of course? We need to understand which parameters have a biggest impact of our predictions in our models. How does it work? Individual parameters are systematically varied, so such as hormone production rates or receptor binding affinities, and model responses are observed, which changes have the biggest effect on plant growth. And the critical components are identified. It helps prioritize which biochemical processes need further environmental validation. Why is sensitivity analysis important? Because it shows which model assumptions are most influential. It helps refine experimental tools, reducing unnecessary lab work. It improves model reliability by testing its stability under different conditions. After, of course, identification key parameters, we need precise value of them. How do we estimate unknown parameters? It's important by using the least square regression, mathematical statistical analysis, fits the model to experimental data by minimizing the difference between prediction and observation. The second, Bayesian interference, use probability to refine parameters estimate based on prior knowledge and new data, and machine learning techniques, neural networks, and optimization algorithm which can improve model accuracy by analyzing large data sets. Why is parameter estimation important? Because fill in gaps where direct experimental measurements are impossible, improves model accuracy, making predictions more biologically relevant, and then prevalent overfitting, ensuring model work beyond the data set they are trained on. Even with the best model validation, it's never perfect. Next, we must know about the biggest challenges in model validation and how we can overcome them. It's very important. Imagine trying to predict. We know that weather sometimes even with the best technology. Imagine predicting the behavior of living, growing, adapting plants. That's the challenge the next challenge of model validation. Why is it so hard? Because plants are unpredictable. Two plants with the same genetics can still behave directly due to the microenvironmental variation. Some data is missing. Scaling up is stopped. A model that works for a single cell may fall when applied to entire plants. How to improve validation? We must run models under multiple conditions to test their adaptability, use machine learning to fill in data gaps and refine predictions, and commonly compare with new experiments to adjust and improve accuracy. To the end of the day, models don't need to be perfect, but they must be useful, helping us understand plant better and development more resilient crops. Where is phytohormone modeling headed? The field of phytohormone modeling is evolving rapidly, driven by advancement in data science, imaging, and computational power. So, what's next? Integration with high throughput omics, real-time modeling with AI, advanced imaging techniques, for example, fluorescence lifetime imaging microscopy, and super resolution microscopy will provide unprecedented insight into hormone movement and interactions. Why does this matter? More accurate models will help us predict plant responses to climate changes. AI-driven modeling can optimize breeding strategies for stress-resistant crop. Multi-scape integration will allow precision agriculture, where models guide real-time decision-making for farmers. The future of phytohormones modeling is a fusion of biology, artificial intelligence, AI, and high-tech imaging, pushing us closer to a new area of precision plant science. Let's dive deeper into how AI is transforming real-time hormone modeling. Imagine if we could predict how a plant will grow before it even happened. That's the promise of AI-driven real-time modeling in phytohormone research. How does AI improve modeling? By instead predictions, AI can process massive data sets and predict how plant response will respond to the genetic or environmental changes in real time by pattern recognition. That means that deep learning models identify hidden correlation in hormone signaling that human might miss. And by adaptive simulation, unlike traditional models, AI can continuously update itself, refining prediction as a new data became available. What does this matter? Help optimize genetics modification to enhance plant traits, enable early stress detection, and improve precision agriculture, where hormone-based model guide crop management decision. As AI continues to advance, we are moving towards a future where plant growth can be simulated, optimized, and ever controlled with unprecedented precision. So, how advanced imaging technology is enhancing model accuracy? To improve the accuracy of phytohormone models, we need better data. And that where cutting-edge imaging technology come in. This tool allow us to see hormone in action, capturing the movement and the reaction with unprecedented detail. Key imaging transformation hormone modeling are fluorescence lifetime imaging microscopy, I said about it, confocal and super resolution microscopy, and biosensor of lime cell living. This is a new molecular probes allow scientists to measure hormone fluctuation in real time, improving model accuracy. What does this matter? More precise imaging, reduce error in computational models, help confirm how hormone move, interact, and respond to environmental changes, and enables new data-driven improves in hormone-based crop breeding and stress adaptation. By combining high-resolution imaging and AI and computational modeling, we are entering a new and precision plant science, where we can see, predict, and manipulate plant hormones dynamics with extremely accuracy. Thank you very much. 1 hour. Yeah. I'm tired. Thank you.